inside the mitochondria. There's a preparatory step where we have the connection of glycolysis to the citric acid cycle, and it's where pyruvate is converted to this 2-carbon acetyl group, and where the first carbon dioxide is released. So let's take a look at the overall reaction of what happens inside the mitochondria, where we have pyruvate with 2-acetyl-CoA, and then NAD plus is reduced to NADH, and the pyruvate is broken down into a 2-carbon molecule that attaches itself to this stuff called coenzyme A, making acetyl-CoA, and 2 carbon dioxides are released. If you remember, carbon dioxide is a small nonpolar molecule, and it easily diffuses out of the mitochondria membrane and out of the cell, and eventually is exhaled as carbon dioxide. And so we have these 2-acetyl-CoA, and they are going to go inside the mitochondrion. So let's look at the mitochondria. The anatomy of this is important. Remember that mitochondria probably had a very ancient origin as a bacterium, and through endosymbiosis became part of a eukaryotic cell. You'll see that the mitochondrion has a double membrane, a rather smooth outer membrane, and then this convoluted inner membrane called the cristae. And it is in the membranes of the cristae that we have the electron transport complex. The mitochondrial matrix is the very inner part of the mitochondrion, maybe the cytoplasm of that ancient bacterium, and that is where the citric acid cycle components are found. And so the citric acid cycle happens inside the mitochondrial matrix, and then electron transport occurs on the membrane surface of the cristae. Another name for the citric acid cycle is Krebs cycle, because a guy named Krebs discovered it. We are reverting to more descriptive terms in biology, and so citric acid cycle is the name because citric acid is the 6-carbon molecule that is formed when the 2-carbon acetyl-CoA joins the 2-carbon part of that to a 4-carbon molecule oxaloacetate, making citric acid. During the citric acid cycle, we also have those coenzymes NAD plus and FAD being reduced to NADH and FADH2, and ATP being formed by substrate level phosphorylation. Not a lot of ATP. And so the citric acid cycle will turn twice for one glucose molecule, one for each pyruvate. And we saw a cycle before when we looked at photosynthesis, and that was the Calvin-Benson cycle. Here is another cycle, the Krebs cycle, or citric acid. And so the end products of the citric acid cycle are 4 carbon dioxides, 2 ATPs, 6 NADHs, and 2 FADH2s. We're not talking about the preparatory step where we had another 2 carbon dioxides made. So let's take a look at an animation of the citric acid cycle.
The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain,
which we'll do after we look at this in a lot more detail. So here we have an overview of the citric acid cycle and where it happens. And you'll notice that we have many compounds named in through here. That's going to be important later when we talk about proteins. The acetyl-CoA is going to be important when we talk about fats later on. So you see that the citric acid cycle starts when acetyl-CoA transfers its 2-carbon acetyl group to oxaloacetate, forming citrate. And so here is what happens here. It regenerates coenzyme A that can pick up another acetyl group from a pyruvate later on. The citrate then gets rearranged and actually reduces NAD to NADH2, and carbon dioxide is released. And so we now have a 5-carbon molecule called ketoglutarate. And the ketoglutarate then causes NAD to be reduced once again, and another carbon dioxide is lost, and an ATP is formed. And so this ATP is when the energized phosphate is transferred from a substrate here to ADP. Now there are other intermediates here that we're not going to talk about. We're just looking for 6-carbon to 5-carbon to 4-carbon. If you study this later or in biochemistry, you will learn each step, and each one of these has a specific enzyme that allows the reaction to occur, that catalyzes the reaction, that lowers the activation energy required so that the reaction can occur. And then FAD is involved here when succinate is oxidized to fumarate and FAD is reduced to FADH2. And then when fumarate is oxidized to oxaloacetate, NAD is reduced to NADH. So we have 1, 2, 3 instances of NADH being made, 1 instance of FADH made for each turn of the citric acid cycle. We have an ATP made for each turn of the citric acid cycle and 2 carbon dioxides for each turn of the citric acid cycle. And here is the tally of the input, the output. You'll see that we started with 4 carbons but in a different arrangement. They were in 2 carbon groups. Now we have carbon dioxides. We have NAD and FAD being reduced, and we have ATP made. So for each of the glucose molecules that started out with glycolysis, we end up with 2 acetyl groups going into the citric acid cycle. So far we've only had a total of 4 ATPs being made. So you know, it's like, okay, so we had 2 made in glycolysis, citric acid cycle 2 made. What's the big deal? Where are we going to